Joanne, you should have received an invite and you're here. Hi, thanks for joining us from the UK. This is late for you, but thank you for being with us. And we all are very, very much looking forward to learn more about your work. I have had several of my students use Cell Typist, and we oftentimes compare results with that results that comes from Asimots or from other cell annotation tools. And it's really interesting for me to learn more and more about how these cells are named. And I think many of our general audience members are interested to learn more about that. So if you want to share your slides and tell us a little bit more about Cell Typist, that would be wonderful.
Yeah, thank you. Yeah, I will start to share my screen.
And Joanne comes from the Sanger Institute in the UK, very famous institute with lots of high-powered experts.
Yeah, thank you. It's really nice to be here. And this is Chuan Xu. I'm a postdoc from Sarah Teichmann's group at the Wellcome Sanger Institute. It's a great pleasure to have this opportunity to share with you some concepts in terms of naming cells using gene expressions, as well as to share with you our work towards this direction by classifying and naming cells with a machine learning approach, which we call Cell Typist. So first, why do we need to name cells using gene expressions, and how does this approach contrast with others? The answer to this question may probably be found in the central dogma of biology, where the genetic information in the DNA is selectively transferred to RNA, followed by protein synthesis and functioning in different contexts, leading to our discovery of different cell types distributed throughout the human body. For these cell types, although we can already distinguish their identities to some extent by cell properties such as morphology, connectivity, and functional rules, there is still a need to explore a more fundamental characteristic contributing to the differences among cell types. And because DNA sequence is nearly identical between any two individuals, as well as between two cells within an individual, tracing back this information all the way to RNA may represent a more fundamental way to classify and name cells. Here shows an example from our previous work where a very specific migratory dendritic cell can be anchored using single molecule fluorescence in situ hybridization. And in this example, co-expression of three important genes, ITGAX, AIRE, and CCR7, uniquely points to its presence in the human lymph node. And this is a relatively simple example because we classify, name, and validate this cell type with only three genes. This may suffice in some cases, but the challenge here is that not all cell types can be uniquely defined by a handful of genes. So to overcome this, a natural solution you may come up with is to make use of all genes within a single cell. In other words, leveraging information of more than 30,000 genes in the human genome may provide more insights into the identity of a given cell. And this is where machine learning or deep learning come into play, in a sense that specific rules will be learned by the machine. And based on this, a cell of interest can be matched against a database of cells to fetch its identity using more than 30,000 genes. And one thing to note here is that although using more genes can help identify cell identities and cell type annotations, the so-called curse of high dimensionality will show up in the meantime. Specifically, if you only have 2 genes expressed in cells, you can easily play around with this 2-dimensional space and to get a picture of how these cell types manifest in this space. In this ideal example, the 2 genes are overexpressed in the 2 cell types respectively. And further leveling up this space to 3 dimensions, you will, as you see, begin to have some difficulty manipulating these cell types, as well as to learn a rule that can be used to separate these cell types. And finally, when all genes are represented simultaneously in this space, and finally for a multi-dimensional space, a machine learning classifier that can be used to demarcate these cell types will be in urgent need. And cell typist, which is developed by our lab, actually is designed to tackle this problem. Here shows an example of 2 cell types staying in a 3-dimensional space, but the general principles that will be mentioned below are applicable to more cell types and higher dimensionality. And for cell typist, we built it on a logistic regression framework, meaning that we are trying to find a plan that can be used to separate cell types, or a hyperplan, a jargon used in a high dimensional space. And with this plan, when an unknown cell enters this space, we can not only predict what cell type does this cell belong to, but also tell how confident our prediction is by considering the relationship between this cell and this plan. And a very important consideration during this process is that how robust this plan is. A very practical and straightforward solution is to have more high-quality cells, or in other words, more high-quality data points. And when the robustness of this classifier is improved, we are able to predict more cells and more cell types in a more confident manner. And the cell typist utilize these 2 information, including a logistical regression classifier, as well as a collection of cells and cell types. And between these 2, I want to emphasize the second part of here of data-driven, as this notion stands for a data-driven framework. And because in humans, there exists a variety of cell types distributed across a range of organs, with their status being influenced by different conditions. And such a dynamic and integrated network signifies that a lot of circumstances should be tackled, such as rare cell types, very homogeneous cell types and subtypes, as well as tissue-shared and tissue-specific cell types. So in summary, in order to classify this cell correctly, theoretically, you need to have a good command of all this cell type knowledge, which is actually the very usual approach people will adopt in daily life. That is, applying their knowledge to unknown cells and deriving their identities manually. And in recent years, automatic cell type annotation is made possible through supervised or semi-supervised methods, which perform the task of label learning and transferring from annotated to unannotated data, transforming this task from knowledge-driven to data-driven. And between these 2 aspects, single-cell genomics is no exception under the general framework of machine learning. And specifically, when you have limited data, you have to rely much on the knowledge, and you have to model the data extremely careful, such that the derived model can be well extrapolated. And on the other hand, when you have more data, the demand for the knowledge will be correspondingly reduced. In an extreme case, when you have sequenced all the cells in the world, you can just have a lookup table and do not need any machine learning methods to predict cells because there are no unknown cells in the world at all. And fortunately, we are at the era of more and more single-cell datasets being generated and raising the possibility that the model can be well generalized at a low cost of model selection and tuning. And the combination between a cross-tissue database and server and an interpretable cell type label projection pipeline, the combination of them can largely ease the challenge of cell type identification and leading to our development of cell typist. And as an example, here shows the 19 datasets we have collected in order to build a cell typist model focusing on immune cells. Immune cells only represent a fraction of cell types in the human body. So you can imagine that we may need to collect more datasets to build a more comprehensive model in order to cover more cell types. And why is this necessary? Because this is the power of big data. When we collected more cells for a given cell type, its prediction accuracy will be improved, as shown here in the y-axis, and converged to a high level. This is actually consistent with previously mentioned trade-off between cell type knowledge and the amount or volume of your data. And continuing with our immune cell example, after we compiled these datasets, we have a dedicated endeavor to build a separate panel for looking up these cell types in a single place. And within this so-called immune cell encyclopedia, we enumerate all cell types we have collected. And for each cell type, we have detailed meta-information, such as cell type description, and curated marker genes, tissue of origin, as well as marker gene expression across tissues. So all this means that you can retrieve different kinds of information, all relating to a single cell type. And given this large database, the next question is, how can we make full use of them? So based on this large database, we are able to produce a suite of cell typist models. These models have two advantages. First, each model is portable and on average 2 megabytes, making cell predictions and classifications quite easy and quick. And second, these models are very comprehensive, covering 101 immune cell types and more than 100 other non-immune cell types from different tissues such as lung, kidney, and liver. And till now, you may still wonder, what does a cell typist model mean in this context? So going back to the previous illustration, a cell typist model is actually a collection of hyperplanes that we provided to the user, which can be used to predict unknown cells. Because we only provide this plane or these models rather than all data points, cell type annotation or classification can be achieved in a lightweight manner. And even when the users do not input all genes, cell typist can still utilize as much gene information as possible. And in practice, we have used cell typist to annotate different kinds of datasets, such as the one shown here, comprising more than 300,000 immune cells collected from 17 human lymphoid and non-lymphoid organs. With cell typist, we are able to resolve its heterogeneity within this dataset to an expert level. Notably, many cell types are very similar to each other, and they often form a continuum in their transcriptomic space. Cell typist, however, can still untangle this phenomenon to a greater detail, such as different T cells shown here with different functional and effect phenotypes, as well as to identify very rare cell populations, such as very specific dendritic cells in the myeloid compartment. And in addition, due to the robustness of cell typist, it allows its application in datasets with different sequencing or sample preparation protocols. In these datasets of SMART-seq2, PEGs, and Cy-RNA-seq3, representing rich, intermediate, and sparse gene expression levels in individual cells, respectively, the prediction result from cell typist matches well with the original cell type annotations provided by the original studies. And even some cell types that are not well characterized in the original study can be further refined, and showing the power of cell typist in further refining existing annotations. And currently, cell typist is already available as a web service. And using this online service, you can transfer cell type labels from the different cell typist models to your data of interest, and to search for information of your predicted cell types. And cell typist is also available in GitHub, as well as a Python package. And in the future, we plan to expand the cell typist database and its models to other scenarios, such as cells on the particular developmental lineages or cells from other cell lineages. And that's all about my presentation. To summarize, classifying and naming cells using gene expression can more faithfully capture the cell properties compared to other cellular features. And using gene expression, this is a better way to represent cell identities. And using machine learning approaches such as cell typist, we seek to leverage the entire gene programs for cell and cell type annotations. And last but not the least important, cell typist also features the cell type database we have collected thus far with different levels of resolutions. And we have continuous updates to further communicate with the single cell community. And finally, there are a lot of people to acknowledge here. This work is guided by my supervisor, Sarah Techman. And I work with Cecilia and Tomasz on this project. And there are important inputs from the cell typist annotation team and many others I may not enumerate here for the important support and feedback. I also want to thank Martin for the technical support, and to Maria, David, Alex, Alan, and other members of the cell ontology team. And thank you for your attention.
Thank you so much. That was a great presentation. And I think there are a number of questions in the chat. You might have seen them. So Ellen Cordocus is asking, how can you tell the difference between different cell types versus cell states of a particular cell type?
So taking the immune cell database as an example, when we collect the old data sets and try to distinguish between different cell states, we adopt a safe way, which means each cell type is well recognized by the community. And maybe some cell types we can further split them, but this split is not so popular in this field. Then we still decide to not split them. So for this database, we try to make it safe. So that's how we differentiate between cell states and cell types that make it safe. Yeah.
And maybe I have to add to this. So what do you do if two experts can't agree with each other? Do you bring in a third expert?
No, you worry too much. Finally, one will agree with the other. So, yeah.
Very good. The other question that came up was, we just had a panel on antibodies and proteins, and protein can also be used to characterize cell types. So if you now take gene-based cell type annotations and compare them to protein-based cell type annotations, for the very same set of immune cells, what differences would you expect to see?
So I think, so when you only use gene expression to predict cell types, there are you can borrow some evidence from the proteins. So, but when you have more genes, the value of the protein is not so high. So I don't mean that the protein is not important. I mean, like when your gene expression is sequenced to a very good level, and then your reliance on the protein will be reduced. But the protein is still useful, especially for many immune cells, such as their TCR and BCR data to find some phenotypes. So actually, previously we had a plan to combine gene expression and protein expression to have some joint classifier, but this should need some assigning weights between the gene expression and the proteins. So we have such plan, but now for current cell types, it's still not possible to combine these two features.
I have one more question, and there might be some from the audience also. It would be wonderful to have a gold standard, fully annotated data set that could be used to compare different cell type annotation tools. Are you aware of any gold standard like it, or the development of such a gold standard?
So do you mean how to compare similar tools?
Yeah, so there are many different cell type annotation tools. So for instance, there is Cell Typist and there is Asimots, which is widely used in HubMap right now. And it would be wonderful to compare them. But if you do that, you get different results. In some cases, they agree. In other cases, they don't. So in order to resolve discrepancies, it would be good to have a fully annotated gold standard data set where experts have spent a lot of time to basically manually annotate cells. For segmentation challenges, we have these kind of data sets. It's very expensive to create them. It's non-trivial to create them because you want multiple experts to all agree on the outlines of single cells or functional tissue units or anatomical structures. So it's time consuming, and you need experts that are typically very, very busy and very expensive to get. But I wonder if such a data set is under development for cell type annotation. So again, oftentimes you want the outline of a cell, but you also want the name. You need both. In order to create a map or an atlas of the human body, you really need a name and an outline in 2D, ideally 3D, for major cell types, anatomical structures. And so I wonder if your team or others in Europe or anywhere on this planet really are working on such a gold standard.
As I mentioned in the slides, we put a lot of efforts to curate and to these datasets. And we not only provide an algorithm for cell type classification, but also provide a high quality database. So people can use this database for different purposes, including cell typist models. Actually, our models and database are continuously updated, and we have efforts to add more cell types or to expand the size of this cell type, means the number of cells within this cell type. So our database is continuously updated, and we try to have some golden standard, but we hope that our database, the quality of this database can be quite high. Yeah.
Very good. Thank you for all that effort. I think it's really needed to have these gold standards so that existing algorithms and new cell type annotation methods can be benchmarked and can be compared and can be validated against these golden standards databases. Another question that always comes up in HubMap is, what data set is good enough to be used for atlas construction as opposed to data sets that can be mapped onto the atlas, but might not have the qualities that you would really like to have in terms of rigor and biosignal over tech noise, so that they should maybe not be used for atlas construction. And of course, as technologies evolve, as new data sets come into existence, you might like to discard some of the old data sets that were used for the early versions of the human reference atlas or human cell atlas and replace those by new versions. Like, for instance, the lung map team has created beautiful data, and the first Asimots reference used the initial version 1 data set, and now Asimots uses the version 2 data set. So I think over time, we will see that certain data sets become obsolete and new ones get created that are much higher quality and have much more cells so that you get more statistical power also. Can you tell us a little bit more about how you pick data sets that you use in your references for cell typist?
So the overall idea is to have more data sets to control for some difference between individuals and the difference between different technologies. And when you have a very heterogeneous database, you can handle different kinds of input data sets. And after collecting these data sets, we also have some selection or some quality control for these data sets. For example, removing some low quality cells from the initial data sets. So overall 2 points. One is to have more data sets, and the second is to keep high quality cells. And yeah.
Very good. There's another question from the audience. Have you compared cell typist and Asimots?
Yeah, I have. In our manuscript, we have compared cell typist with different tools, and the performance of cell typist is better or comparable with most tools. And one thing very important, I think, is sometimes the quality of the reference atlas is more important than the algorithm you used. When you have a bad reference, whatever algorithm you use, the result will not be good, and vice versa. So, yeah.
Excellent.
Overall, overall, cell typist has good performance. Yeah.
If you can post the link to that algorithm comparison, I think there are people in the audience that would love to get the details. Thank you so much, Xuan, for coming in on such a late time for anyone in Europe. Really good to see you. And I will go over and introduce San Fang. She is from Johns Hopkins University. She's actually part of the HubMap team, part of the other mapping center, Rahul Zahid's team at the New York Genomics Center. But Jan Fan is actually at Johns Hopkins University. Jan couldn't join us today. She's actually traveling in Germany right now. And she shared with us some recordings that I think all of you will find most interesting and educational. So one of them is on spatial transcriptomics. I think that's the first one you get to see. And I'm going to share this here. And then, Ellen, if you can make sure that I actually get the sound right this time, that would be wonderful. All right, I'm sharing the video and I'm starting it.
What is spatially resolved transcriptomics?
Spatially resolved transcriptomics refers to a set of new technologies that are enabling scientists to essentially take pictures of cells inside tissues. But rather than these pictures being of how a cell looks, these pictures capture what genes are being expressed in these cells in that moment. By quantifying the different amounts of these genes that are being expressed in each cell, scientists can tell the type or state of each cell and get some sense for how the cell behaves and what it's up to. Using spatially resolved transcriptomics, we can now do this for thousands of different genes and individual cells at a time.
Interesting. But why is spatially resolved transcriptomics useful?
Cells exist in a community inside your tissues. Different cells talk to one another, in part by expressing different genes, thereby influencing each other's behaviors. By measuring what genes are being expressed in each cell, while also looking at how these cells are organized in both healthy and diseased tissues, scientists may be able to better understand how cells are working together to maintain healthy function, but also what goes wrong in disease.
Got it. So will spatially resolved transcriptomics cure all diseases?
We're still in the very early days, so it's hard to say for certain what the future has in store. But many scientists are hopeful that spatially resolved transcriptomics will help lead us to a better understanding of diseases that may in turn generate testable hypotheses and contribute towards a cure. In working towards this future, more scientists and resources are still needed to continue improving these spatially resolved transcriptomics technologies, apply them to various healthy and diseased tissues, as well as develop new statistical and computational methods capable of making sense of all of this rich big data. So if you're a student interested in learning more about spatially resolved transcriptomics and even contributing to this new exciting scientific field, please check out the JeffWorks Lab website and apply to do an internship with the JeffWorks Lab. We look forward to hearing from you, and we hope you've learned a little bit about spatially resolved transcriptomics in this video.
Thanks for watching.
The human body is composed of trillions of cells. These cells express different genes that enable them to carry out specialized functions. But how are these genes organized within each cell? Knowing where a gene tends to be present within a cell can give us clues about its function and even the future fate of the cell itself. Recent advances in spatially resolved transcriptomic technologies, including multiplexed error-robust fluorescence in situ hybridization, or MERFISH, can now allow us to essentially take pictures of where genes are present within fixed cells. And we can do this not only for one gene or two genes, but potentially thousands of genes. And we can do this not only for one cell, but many thousands to millions of cells. Given this rich data with some clever computational analyses, we can now begin asking questions like, how are genes organized within cells? And more specifically, what genes are located to different subcellular compartments? As a proof of concept, we first sought to focus on a specific subcellular compartment, the endoplasmic reticulum. The translation of genes that encode secreted, glycosylated, and/or transmembrane proteins, collectively termed the secretome, has been shown to take place on the endoplasmic reticulum. Therefore, we have some expectation that these secretome genes should be spatially co-localized to the ER. To test whether our expectations are correct, we combined Murfish spatial transcriptome profiling with KDEL immunolabeling, which marks the endoplasmic reticulum. We computationally segmented the regions of our image corresponding to the endoplasmic reticulum for each cell, highlighted here in red, as well as the regions outside the endoplasmic reticulum for each cell, highlighted in blue. And we counted how many copies of each gene we observed within each cell's ER and non-ER compartment. We then performed a pairwise inferential statistical analysis to identify which genes were significantly more enriched within the ER compared to the non-ER compartment. As shown in this volcano plot, each point is a gene, and genes in this upper right quadrant are significantly enriched within the ER compared to the non-ER. We then highlighted in red our gold standard secretome genes, which we had expected to be enriched in the ER. And indeed, we see a large number of these genes as being significantly highly enriched in the ER compartment. Further, as Murfish is an imaging-based approach, we can actually visualize all our identified significantly ER-enriched genes on top of the original KDEL staining image. If we zoom in, we can indeed see that these genes are beautifully spatially localized to the fine networks of the single cell's rough ER network. Having demonstrated that our approach is able to recapitulate the expected known biology regarding the spatial localization of secretome genes, we next sought to explore a less well-characterized compartment, the nucleus. Applying a similar approach as previously, we were able to identify a large number of genes highly enriched within the nucleus. Of these genes, some were long non-coding RNAs, which are known to be localized to the nucleus. In addition, RNAs with retained introns were also preferentially enriched in the nucleus as expected. Interestingly, although protein-coding mRNAs are translated in the cytoplasm, we identified a large number of protein-coding genes to be highly enriched in the nucleus. A number of functions have been proposed for the accumulation of a mature protein-coding mRNAs in the nucleus. For example, retention of mRNAs in the nucleus may allow cells to quickly respond to stimuli by exporting these retained RNAs to the cytoplasm to upregulate translation, thereby bypassing the slower transcription step. In this manner, scientists, including those in my research team and others, are enhancing our understanding of how subcellular organization relates to cellular function and heterogeneity. For more details on these research findings and more, please check out our original scientific publication in PNAS, also linked below. My name is Jean Fan, and I'm an assistant professor in the Center for Computational Biology in the Department of Biomedical Engineering at Johns Hopkins University. When we look at a complex organ like the mammalian brain, we may be interested in asking questions like, how are cell types and cell states spatially organized within this tissue? And how does this spatial organization change during healthy development, as well as the progression of disease? By addressing these key biological questions, we may be better able to understand how spatial organization plays a role in tissue function, as well as disease pathogenesis. In order to address these interesting biological questions, it becomes very helpful if we could make precise quantitative measurements regarding the molecular features of cells within the tissue while preserving their spatial organizational information. Recently developed spatially resolved transcriptomics technologies now provide us with an opportunity to begin addressing these interesting biological questions by providing us with the tools to make these large-scale, high-throughput gene expression measurements of cells or small groups of cells within tissues while preserving their spatial organization. There are many types of spatially resolved transcriptomics technologies. Some, based on spatially resolved capture and sequencing, provide us with full transcriptome untargeted gene expression profiling within small groups of cells within pixels. What these technologies generally allow us to do is take thin tissue sections, such as the one seen here, and allow us to capture the RNAs present at each spatially resolved pixel. By tagging each spatial pixel with a unique barcode and subjecting the resulting libraries to sequencing, we can align the reads to a genome, resolve the original spatial barcodes, and get a quantification of the gene expression at each spatially resolved pixel. Therefore, the process information we end up with comprises the spatial coordinates of the pixels, as well as the gene expression observed at each pixel. As seen here, 2 example genes are shown, and already visually we can begin appreciating how some genes are more highly expressed in some pixels, as noted in red, than others, and different genes will exhibit different spatial patterns due to the underlying cell types and underlying tissue-level processes. We can take all of these gene expression measurements and perform clustering analysis to try to identify pixels that are transcriptionally similar to each other, visualized here as a UMAP, or using additional community detection algorithms, cluster them into putative clusters, which we can visualize both on the UMAP as colors, as well as on the original spatial coordinates. In this case, our clustering analysis allows us to identify transcriptionally similar groups of pixels. However, it's worth keeping in mind that each spatially resolved pixel may contain multiple cells, and as a result, these cells may comprise multiple cell types. Therefore, the gene expression measurements we observe may be a reflection of these cell type mixtures. And subsequently, the transcriptionally similar groups of pixels or clusters we identify through our clustering analysis may actually comprise mixtures of cell types rather than one true underlying cell type. And further, the spatial patterns we observe from these transcriptional clusters may also be confounded by cell type mixtures and subsequently hide potentially finer cell type specific spatial organizational patterns. So we sought to develop a computational method that could help us recover these cell type specific spatial organizational patterns and cell type specific gene expression profiles from multicellular pixel resolution spatially resolved transcriptomics datasets, all in a way that did not rely on external single cell transcriptomics references. So we developed an approach called SD-deconvolve that was recently published in Nature Communications, and you can check out the paper for more details. But briefly, SD-deconvolve builds on latent Dirichlet allocation, a machine learning model popular in natural language processing. In the context of natural language processing, we can think of each document as a collection of words. Of course, intuitively, these words comprise underlying topics that are latent and not observed. However, we can think of these topics as distributions over words. So by observing many documents, each comprising many words, we can infer the underlying latent topics and therefore estimate the proportion of each topic in each document. By analogy, I hope you can see how each spatially resolved pixel is comprised of a collection of genes. Just as how documents are comprised of underlying latent topics, these spatially resolved pixels are also comprised of underlying latent cell types. And just as topics are made up of distributions of words, we can also think of these cell types as made up of distributions of genes. So by observing many spatially resolved pixels, each with different distributions of genes, we can infer the underlying latent representation of different cell types that must comprise each spatially resolved pixel. In this manner, given multicellular pixel resolution gene expression information, SD-deconvolved is able to build on latent Dirichlet allocation to deconvolve back out the cell type proportional representations in each pixel, as well as the cell type specific gene expression profiles. As a proof of concept, we first sought to simulate multicellular pixel resolution spatially resolved transcriptomics data by taking single cell resolution spatially resolved transcriptomics data and aggregating the gene expression within simulated multicellular pixels. Using the simulation approach, we now know the ground truth cell type proportional representation in each multicellular pixel, as well as the cell type specific gene expression profiles. We can then apply SD-deconvolve and evaluate how well we recapitulate both the ground truth cell type proportions and the cell type specific gene expression profiles. And visually, you can see that we do quite well, but we can also quantify this by looking at the correlation between the deconvolved gene expression profiles and the ground truth gene expression profiles, as well as the deconvolved cell type proportional representations and the ground truth proportional representations of cell types across pixels. And in both cases, we see a very high correlation, suggesting that we're able to successfully recapitulate the ground truth. We can also analyze real multicellular pixel resolution spatially resolved transcriptomics datasets, such as this of the mouse olfactory bulb, where we found that clustering analysis alone was unable to recapitulate known subpixel resolution cell type specific spatial organizational patterns, such as the rostral migratory stream in the middle of the granule cell layer, which is not apparent from clustering analysis alone. Applying SD-deconvolve, we were able to identify a particular cell type that spatially was localized towards the middle of the granule cell layer where we expected this rostral migratory stream to be present. And when we look into what genes are upregulated in this deconvolved cell type, we indeed find no neuroprogenitor cell markers. And likewise, when we look to see where these genes are expressed using alternative molecular profiling methods such as FISH, we see that they indeed localize to the interior of the granule cell layer as expected. So in this manner, we find that deconvolution with SD-deconvolve is able to recover the cell type proportions and cell type specific gene expression profiles of neurons within the rostral migratory stream in the mouse olfactory bulb in a way that clustering analysis was not able to reveal. We're able to apply SD-deconvolve to recover cell type specific gene expression profiles and proportions across a wide variety of multicellular pixel resolution spatially resolved transcriptomics datasets and technologies. Returning to our original example, we can apply SD-deconvolve to recover the cell type specific gene expression profiles and proportions, and do this for the whole tissue section to reveal interesting new spatial patterns regarding cell types not present through clustering analysis alone. So with that, I hope you've been able to learn a little bit about what is multicellular pixel resolution spatially resolved transcriptomics data and how we can analyze this type of data with approaches like SD-deconvolve to recover cell type specific gene expression profiles and cell type specific spatial organizational patterns. If you're interested in learning more about SD-deconvolve, please feel free to check out our lab website as well as lab blog for additional tutorials. Cancer is a disease marked by the uncontrolled proliferation of abnormal cells within the body. While cancer can impact a variety of different tissues and cell types within different patients in different ways, even within the context of an individual tumor within an individual patient, subpopulations of cancer cells may exhibit distinct genetic alterations, transcriptional states, and spatial organization, and more. This is called intratumoral heterogeneity. Intratumoral heterogeneity is an important clinical determinant of patient outcomes. Different subpopulations of cancer cells may differentially interact with cells in the tumor microenvironment, as well as with therapies to drive resistance and relapse. Scientists, including those in my research team and others, are using the latest, most cutting-edge molecular profiling technologies, such as single-cell DNA and RNA sequencing, as well as developing new computational methods and statistical approaches for analyzing this type of data, in order to characterize this heterogeneity and delineate the mechanisms by which this heterogeneity may play a role in driving cancer pathogenesis and prognosis. In order to better understand how genetically distinct subclones may differentially shape therapeutic resistance, interact with cells within the tumor microenvironment, and impact clinical outcomes, we need to be able to assess how these different subclones may differ transcriptionally. That is, we need to be able to simultaneously profile genetic and transcriptomic features from the same single cells, from the same tumor, from the same individuals. In order to achieve such simultaneous genetic and transcriptomic profiling in the same single cells, we can either take a technology development approach where we develop new technologies that allow us to measure both genetic and transcriptomic information from the same single cells, or we can take a computational approach where we develop new computational methods that allow us to connect genetic and transcriptomic information from individual cells using existing data. Today, I will focus on briefly discussing the computational approach, but you can always check out either of these papers below in the description for more detailed information. As detailed in our paper, Linking Transcriptional and Genetic Tumor Heterogeneity Through Allele Analysis of Single-Cell RNA Sequencing Data, published in Genome Research in 2018, we developed a computational method called HoneyBadger to infer some genetic information directly from single-cell RNA sequencing data. The general intuition that motivated our computational methods development is the idea that we can infer some genetic information directly from single-cell RNA sequencing data. Notably, single-cell RNA sequencing data is comprised of sequencing reads from expressed genes that may harbor mutations. So in theory, if a mutation is present within the exon of an expressed gene, we should be able to detect the mutation and use these mutation patterns to distinguish genetically distinct cancer subclones. However, in practice, sparse coverage and high monoallelic detection limits our ability to confidently infer somatic point mutations from single-cell RNA sequencing data. Compared to bulk RNA-seq, single-cell RNA-seq has much poorer sensitivity for detecting single nucleotide variants, such as single nucleotide polymorphisms, that we know to be present. Even for highly expressed genes, monoallelic detection, by which only one allele is sequenced, further limits our ability to confidently call heterozygous mutations, which may be present on the non-detected allele. And finally, we simply may not have coverage at all the genomic positions of interest, and in such a case, we would not be able to say whether this cell does or does not have a particular mutation, simply because there is no coverage at that point. In addition to somatic point mutations, cancer cells may harbor genomically larger aberrations, such as copy number variations, or CNVs. In CNVs, chromosomal regions, arms, or even whole chromosomes may be lost or gained. We reason that patterns of heterozygous single nucleotide polymorphisms, or SNPs, present throughout the genome may be used to reveal evidence of such copy number variations in individual cells that could be used to distinguish different genetic subclones. Specifically, even though individual SNPs may be impacted by the sparse coverage and high monoallelic detection as noted previously, joint analysis of multiple SNPs, such as those within regions affected by CNVs, could allow us to confidently achieve subclonal classification. Notably, in bulk RNA sequencing, when we look at the allelic frequencies of heterozygous single nucleotide polymorphisms within copy-neutral diploid regions, we expectedly see a balanced detection of both alleles, as noted in yellow. However, in a region affected by CNVs such as a deletion, we only detect expression from one allele, as noted in blue, because the other allele was, well, deleted. When we profile the allelic frequencies of the same heterozygous SNPs in a population of single cells, the data is understandably a little bit noisier. We can see how SNPs within certain genes exhibit monoallelic detection of one allele, noted in blue, or the other allele, noted in red, and sometimes we can detect both alleles. However, when we look across a large region spanning multiple genes, in a diploid copy-neutral region, we can see detection of both alleles. Whereas in a region affected by a deletion, we see persistent detection from the one remaining allele. To systematically identify and evaluate the probability of CNVs in individual cells from such SNP patterns in single-cell RNA sequencing data, we applied a hidden Markov model to identify putative regions affected by CNVs, and then applied a Bayesian hierarchical model that takes into consideration the sparse coverage and monoallelic detection rates in order to infer the probability of each CNV within each single cell. And we packaged these statistical approaches into an open-sourced R software package called HoneyBadger, available on GitHub, with additional tutorials and resources available on the accompanying software website. In order to demonstrate the utility of HoneyBadger, we first applied it to distinguish cancer cells harboring CNVs from normal diploid cells in a patient with multiple myeloma. Indeed, HoneyBadger was able to identify a number of regions affected by potential CNVs, and further infer the posterior probability of each cell harboring each CNV based on the observed SNP patterns. Hierarchical clustering of the resulting posterior probabilities correctly distinguish the cancer cells, which have high probabilities of harboring the identified CNVs, from the normal cells, which have very low probabilities of harboring the identified CNVs, consistent with what we expect. We further validated the identified CNVs by HoneyBadger using whole exome sequencing, where indeed we confirmed that deletion affected many whole chromosomes, as well as by molecular cytogenetics, where we can see here one dot corresponding to the one copy of MAF on chromosome 16, which is affected by deletion, versus two dots on the two copies of IGH on diploid chromosome 14 as a control. Having demonstrated that HoneyBadger is able to distinguish cancer cells from normal cells based on identified CNVs, we then applied it to analyze multiple myeloma samples collected at two distinct time points from a progressive treatment-refractory multiple myeloma patient. The initial multiple myeloma sample, MM34, was collected from the bone marrow at the time of diagnosis, while the second extramedullary multiple myeloma sample, MM34A, was collected from an ascites dissemination following 2 months of unsuccessful treatment. Applying HoneyBadger to cells from the second ascites dissemination sample, MM34A, we were able to identify a number of clonal copy number variations that affected all cells with a high probability. Interestingly, when we sought to identify these same CNVs in cells from this first bone marrow sample, MM34, while we find that nearly all cells harbored chromosome 13 deletions, for example, only a fraction harbored the chromosome 16 and chromosome 17 deletions, indicative of a linear subclonal expansion, where a genetic subclone harboring deletions on chromosome 13, 16, and 17 most likely expanded to see the extramedullary multiple myeloma dissemination, acquiring additional alterations during this process. We therefore designated this genetic subclone that was most genetically similar to the extramedullary multiple myeloma cells as the extramedullary-like multiple myeloma cells. While we were able to identify key genetic subclones based on distinct patterns of copy number variations, by inferring genetic information directly from single-cell RNA sequencing data, we could then further characterize the transcriptional differences between these different subclones. In particular, we were able to identify over 100 consistently differentially downregulated and also upregulated genes when comparing the extramedullary-like subclone with other multiple myeloma cells in MM34, as well as jointly with the extramedullary multiple myeloma cells in MM34A. Beyond identifying specific differentially expressed genes, we can also characterize what pathways and gene sets are impacted by these differentially expressed genes. Gene set enrichment analysis found that genes upregulated in the extramedullary-like subclone showed significant enrichment in known multiple myeloma partial response signatures, as well as other cell cycle and proliferation-related signatures. In contrast, genes upregulated in the other genetic subclone showed significant enrichment in immune system processes, revealing how these different genetic subclones may exhibit distinct transcriptional signatures that ultimately contribute to cancer progression. Alternatively, we can first characterize aspects of transcriptional heterogeneity among our populations of multiple myeloma cells using methods such as pathway and gene set overdispersion analysis, or PAGODA. And we can then overlay the previously identified genetic information to assess the correspondence between the identified transcriptionally distinct subpopulations with the underlying genetic subclonal architecture. Indeed, we can identify aspects of transcriptional heterogeneity, such as this immune response aspect, that is well aligned with the underlying genetic subclonal architecture. Alternatively, we can also identify other aspects of transcriptional heterogeneity, such as this aspect driven by the expression of chemokine ligands CCL3 and CCL4 that do not appear to correlate with any of the identified genetic subclonal architecture, potentially indicative of other alternative non-genetic drivers of transcriptional heterogeneity, such as the composition of the tumor microenvironment and more. So, I hope we've been able to show you how by inferring genetic subclonal architecture directly from single-cell RNA sequencing data, computational methods such as HoneyBadger can help us better understand the interplay between transcriptional and genetic heterogeneity in cancer progression. And with that, thank you to everyone who has contributed to this research, in particular our collaborators, as well as the funding agencies that made this research possible. And thank you for your attention.
All right. I hope you enjoyed that session. It's wonderful to see what methods now exist to name cells using validated antibody panels that then characterize proteins in cells. But the last session also showed you how you can use gene expression values to name cell types. And again, it's not just names that we need. We also need to understand the spatial dimensions of cells. So the next set of speakers will help us see how cells can be segmented in tissue data, and not just cells, but hierarchies of cells and cell neighborhoods, because it matters in which environment a cell lives. If a cell dies, and the neighboring cells are also impacted. If a cell is well off, then the other cells also benefit. And oftentimes cells work together to create certain types of function, and they need to really collaborate to make that work in the best sense of the word. And we are all trying to understand not just where these cells are, but also how they function together.